All right, we're going to do the neuromuscular junction, excitation, contraction, coupling, and cross-bridge cycling. Haven't done this in class yet. I started it, haven't finished it. So for you guys who've missed, here you go. And for you guys that I'm going to start doing it next week, a little preview. Let me get my pen started. All right. There we go. Okay, events at the neuromuscular junction, excitation, contraction, coupling, and cross-bridge cycling. What's the neuromuscular junction? Where an axon terminal and a muscle fiber come together. So this is synapse. That's a neuromuscular junction, okay? So we're going to talk about what happens there, and then we're going to talk about all the way to the end of cross-bridge cycling when you actually get a muscle to contract. Okay, so let's remember, what are we trying to do here? We've got we're thirsty, okay? We have sensory input. We see the glass of water. We have a signal that goes to our brain, sensory input through a neuron to our brain. Our brain says, hey, I recognize that. That's water, and I happen to be thirsty. I'm going to tell my arm to go pick up that water so I can drink it. So you send out motor output, a response, to pick up the water. So that motor output goes to the muscle, causes the muscle to contract. You pick up the water, you drink it. So you have sensory input, integration, and motor output, or your response. Sensory input would be the stimulus. This would be the decision. This would be the response. Okay, again, just remember where we are. This is a motor neuron. This means it's a neuron that goes to a muscle. All right, got your cell body, your soma, got your axon, got your myelin, you got your nodes of Ranvier, action potentials going this direction, and we have our axon terminals. This is what we're going to be concentrating on, these axon terminals, this secretory region. You remember what's being secreted? Acetylcholine, a neurotransmitter. Okay, so we've blown this up. Here's our axon terminal, okay? Here's our muscle fiber, muscle cell, okay? Here's the sarcolemma, that little membrane around a muscle fiber. Here you have your axon terminal, your synapse. Where's your synapse? Let's use the word synapse, not on here. Between your axon terminal and your muscle. All right, so first step, your action potential arrives to the axon terminal. So here comes your action potential, arrives at the end. That stimulates voltage-gated calcium channels to open. Calcium flows into the axon terminal. That causes these synaptic vesicles that contain acetylcholine to fuse with the membrane and to release acetylcholine. All right, let's see. All right, so the neuromuscular junction. Usually about midway along the entire length of a muscle, you have a neuron will come down and hit about the middle of a muscle so that you can spread the action potential. Each axon terminal and a muscle fiber come together by a little bit of a space, a little gel-filled space called the synaptic cleft. You have synaptic vesicles that contain neurotransmitters, and in this case it's acetylcholine. There's a bunch of neurotransmitters in your body, but for muscle movement it's acetylcholine. And if you have a neurotransmitter, you have to have some type of receptor. So on the muscle side, on the sarcolemma side, you have acetylcholine receptors. All right, so what happens? Action potential travels down the axon terminal. You release acetylcholine. Why? Because calcium, voltage-gated calcium channels have been opened. Calcium flows into the axon terminal. That causes the release of acetylcholine. That acetylcholine will then cross the membrane, cross the synaptic cleft, bind to the other side to generate another action potential. All right, so here's all those steps. A lot of stuff to talk about. Here comes my action potential. Comes to the end of the axon terminal. Causes the opening of voltage-gated calcium channels. Calcium flows in. Acetylcholine vesicles fuse with the membrane. Acetylcholine diffuses across the synaptic cleft, binds to its receptor. Remember, if you have a neurotransmitter, say acetylcholine, has a particular shape, its receptor has the mirror image of that shape. The receptor binds to, right here, here's the receptor. Now, these are ligand-gated channels. Remember ligand-gated versus voltage-gated? That means in order for these channels to open, something has to bind. The neurotransmitter has to bind. In this case, it's acetylcholine. So acetylcholine binds its ligand-gated receptor, okay, and that allows sodium and potassium to move into the sarcolemma. That change in charge generates an action potential. All right, let's see, let me get over. Did I miss anything? Acetylcholine. All right, there's all your steps, the 6 steps I did in class. Okay, so the last step, in order for this to end, in order for this acetylcholine to quit generating an action potential on this side, you have to get rid of the acetylcholine. And there's 2 ways to get rid of acetylcholine. It'll either diffuse away, see the, just basically float away, or it'll be broken down by acetylcholinesterase. All right, I'm going to do it one more time. Because this is one of those things, this is one of those ordered things with lots of steps. So we're just going to go down, make sure you get these steps. You're just going to memorize it, think about what's happening, watch those videos, watch those animations, and just keep doing this over and over and over in your head until you get it right. Here comes my action potential, which is just your stimulus. It's the movement of sodium and potassium right across the membrane, generating a charge called an action potential. Action potential comes down the axon to the axon terminal. That change in charge, that change in voltage, opens up a voltage-gated calcium channel, allows calcium to enter. Calcium binds with these little synaptic vesicles. They then exocytose or release acetylcholine into the synaptic cleft. Acetylcholine then diffuses across the cleft, binds to its receptor. That receptor is a ligand-gated channel. That opens the channel, allows sodium to enter and potassium to exit. That causes a membrane change, current change on the sarcolemma. This is the sarcolemma. And that generates an action potential. Once your action potential has been generated, you no longer want that signal. You come in, acetylcholine diffuses away, or it's broken down by acetylcholinesterase. Remember, ACE is an enzyme. It just breaks it down. Okay. I wrote this out for you because I know when I say acetylcholinesterase, you're like, how do you spell that? Acetylcholinesterase. All right, that is basically the enzyme that breaks it down. For me, if you have to write this out, it's okay to write ACH. I'll know what you're talking about. Okay, that's acetylcholinesterase. Once it's broken down, there's no more acetylcholine in the synaptic cleft. There's nothing else binding the ligand-gated sodium-potassium channels. No more action potential. That's the end of the stimulus. Okay, now, we've got to generate an action potential in the sarcolemma. We've had an action potential coming down the axon. Okay, we've released a neurotransmitter into the synaptic cleft. Now we've got to generate an action potential on the muscle side. Okay? How does that happen? The acetylcholine, it binds those ligand-gated channels. Okay? And when it does that, these there's some channels, sodium and potassium. They open, more sodium comes in, more potassium leaves. It's usually more sodium coming in. More sodium diffuses in, which causes the inside to become less negative or more positive, and that basically causes a little bit of depolarization, which then generates an action potential. Okay, so more sodium flowing in, the potassium is leaving, causes the membrane potential to change, causes it to depolarize, and you generate an action potential. All right, so here's our muscle cell, muscle fiber. The sarcolemma is that membrane that surrounds the muscle cell, muscle fiber. So this is the sarcolemma that we're looking at. This is the membrane that we're looking at, okay? This is our sarcolemma. Here comes our axon. Here comes our acetylcholine. It diffuses across the membrane, binds the ligand-gated channel, causes a local depolarization, okay? Then that wave of depolarization will then spread all across the membrane. You again have little voltage-gated sodium and voltage-gated potassium channels, just like in the neuron, along the sarcolemma, that are able to carry the action potential all the way across the muscle cell. So, and it's almost instantaneous, so that the entire muscle cell will depolarize, and the action potential will, it's pretty much instantaneous. The whole thing depolarizes at the same time, so that your muscle will contract together as one big unit, okay? So it's very, very quick. So you have to generate it right here by the axon terminal, and then the action potential will spread out all over the muscle. Whoops. Okay. You should remember this from my last talk. For something to depolarize, sodium channels open, more positives rush in, membrane potential becomes more positive, those channels close, potassium channels open, potassium leaves, causing the membrane potential to become more negative, potassium channels close, and you go back to resting membrane potential. So just like what we talked about going on the axon, the exact same thing is going on the sarcolemma of the muscle. Okay. Let me see. Hang on just a second. All right, I'm going to go back. Let me show you some anatomy before I talk about what I was going to talk about. This is the sarcolemma, okay? So this is basically that membrane that's capable of generating and propagating an action potential all the way along a muscle, all right? The sarcolemma will actually invaginate, okay? It kind of goes down into the muscle cell. Remember, these are myofibrils that you're looking at. All right, so the sarcolemma will actually go down inside the muscle cell, and you've got what's called the sarcoplasmic reticulum, which is basically a web all over, right underneath the sarcolemma and over the muscle, the myofibrils. So this sarcoplasmic reticulum is the web, okay, like an endoplasmic reticulum that stores calcium. So what happens is this action potential travels down the sarcolemma, and then it goes down into the cell through these invaginations, which are actually called transverse tubules, T-tubules for short. So it goes down the transverse tubule, down the T-tubule, and actually causes a response in the sarcoplasmic reticulum to release calcium. Excuse me. All right, so let me go back. So just have that in your mind. You have your sarcolemma. It actually kind of goes down inside the cell, okay, meets with the sarcoplasmic reticulum, which is where you're going to release calcium. The whole goal of this next step is to release this calcium. Calcium is the trigger that allows contraction to occur. You've got to have this release of calcium in order for contraction to happen. You remember we talked about calcium. It's not just for your teeth and your bones. It has a whole lot to do with muscle contraction, a whole lot to do with nervous system conduction. We've already seen one time you've got to have calcium in order for a neurotransmitter to be released. You've got to have it. Now we're going to say that you've got to have calcium in order for a muscle to contract. So let me go back. Okay, so excitation, contraction, coupling. Basically, this is where you bring together or couple the electrical event with the mechanical. The electrical event, the action potential, and the contraction. That's the mechanical event. So you're basically you're wanting to convert or couple an electrical event into a mechanical event. You actually want to contract. So there's a lot of sequences, a lot of steps we're going to go through in order to have that occur. Okay, so your action potential goes along that sarcolemma, okay, into the T-tubules, which are called the transverse tubules. Really great picture in your book. If you haven't looked in your book, there's a really great picture of the setup of the sarcolemma, the sarcoplasmic reticulum, and the transverse tubules. On the transverse tubules, you have, again, voltage-sensitive proteins. Okay, they're almost like voltage-gated channels. A little bit different, but for all intents and purposes, almost the same thing. Basically, voltage-sensitive channels that will allow calcium to be released. So when that action potential, here's your sarcolemma, it goes down into those T tubules. When your action potential charge goes down into the down into the T tubule, there's a voltage change that allows calcium to be released. And then once you've done this, you have now released your trigger for contraction. All right. So you got your axon, had all your steps we've talked about before. Action potential comes down, calcium goes in, acetylcholine is released, crosses the synapse, generates another action potential on the sarcolemma, travels down the sarcolemma into the T tubule. Now you're going to get a release of calcium. Now this calcium, that's actually that's the coupler. Once you release calcium, you will get contraction. Okay. Another word that I need to talk to you about is called the triad. If you look at a sarcomere, between the little Z lines and the little Z lines, where one sarcoplasmic reticulum, there will be like a web-like over here, and there's sarcoplasmic reticulum over here. You have a T tubule or a transverse tubule, and then a sarcoplasmic reticulum from this side and one from this side. Where one sarcoplasmic reticulum, a transverse tubule, and another sarcoplasmic reticulum all come together, that's called a triad for 3. So 3 things, 2 sarcoplasmic reticulum and 1 transverse tubule makes a triad. So basically that transverse tubule brings that voltage change, the action potential, down into the cell. That voltage change opens those calcium channels. Calcium then is released into the cytoplasm of the muscle cell. All right. Calcium is now out into the cytoplasm of the cell. Now you're going to get contraction. Here's our little calciums. Do you remember our thin filaments and thick filaments? You have myosin, you have actin, you have the little string holding the actin is the tropomyosin, and this little yellow thing, that's troponin. Troponin is a place for calcium to bind. Troponin has a calcium binding site. Okay? So once calcium is released into the cytoplasm of the muscle cell, it will bind troponin. Okay? When troponin is bound, it actually causes tropomyosin to change shape. It kind of causes it to roll. And when it changes the shape of tropomyosin, these myosin binding sites on actin are now exposed. See how troponin is covering up the binding sites? Calcium binds, the troponin changes shape, and now your myosin binding sites on actin are now exposed, so myosin can now bind. Okay, so once myosin and actin bind, you get contraction. But we're not quite there yet. Hang with me. Okay, so I'm just going to keep saying it over and over and over again. I promise everybody gets it eventually. Just don't pull your hair out quite yet. All right, here comes my action potential coming down the sarcolemma. All right, and as it comes past the T tubule, it's going to go down inside the cell. It's going to cause a voltage change, cause that calcium to be released from the sarcoplasmic reticulum into the cytoplasm of the muscle cell. All right, calcium's released. All right, calcium is then going to, you go to the next slide, bind troponin. Okay, that binding of troponin is going to cause tropomyosin to change shape and remove and quit blocking the myosin binding site. So here you have the myosin binding site is blocked. The tropomyosin basically changes shape, and now you have a binding site that's available for myosin on the actin. When actin and myosin join, that's called a cross bridge. And so if you remember that video we looked at in class where you have the little myosins doing this, those little cross bridges, they're going to keep doing that. No, that looks silly. They're going to keep doing that and shortening that sarcomere, shortening the muscle, and you're going to get contraction. All right. So that was a bunch of steps. Now, there's even more steps involved in this part. This actual cross bridge formation has even more steps you've got to learn about. This is where ATP comes into play. All right. So that's just a kind of review. Put it all together again. Here comes your action potential. Here comes your voltage change. Here comes your opening of your calcium channels. Calcium leaves the sarcoplasmic reticulum, goes into the cytoplasm, binds to the troponin, causes tropomyosin to change shape. Actin and myosin bind, you get contraction. Now we're going to work on this part. All right. Now, calcium, let's see how I'm going to say this. In order for a contraction, you've got to have that calcium, okay? Because it's got to bind that troponin in order to move that tropomyosin. As long as calcium is in the cytosol, as long as it's there, actin and myosin will continue to bind, and you'll still get contraction. You've basically got to remove the calcium in order for the contraction to stop. For you to get muscle relaxation, you've got to remove the calcium. To pump calcium back into the sarcoplasmic reticulum, what do you think that takes? Pump. Takes ATP, takes energy in order to pump the calcium back into the sarcoplasmic reticulum. So as long as that calcium's in the cytosol, this contraction will continue. So we've kind of already talked about two things that you've got to have in order for a contraction to stop. Acetylcholine, as long as it's in the terminal and as long as it's in the synapse, you're still getting a signal, so you've got to get rid of the acetylcholine, okay? So you have to do that in order for muscle to relax. And then now we know you've got to get rid of the calcium, because as long as there's calcium inside the cell, freely inside the cytosol, you're going to get contraction. So you've got to stop the acetylcholine and you've got to get the calcium out in order for a muscle to relax. Can you think of a time that might not happen? Keep thinking about it. We'll get there. Okay. I think this is kind of review what I just talked about. Calcium binds to troponin. Troponin causes tropomyosin to change shape. Cross-bridge cycling occurs. Once you're ready for the contraction to stop, you pump the calcium back in to the sarcoplasmic reticulum. That takes ATP, and the contraction ends. Now we're at cross-bridge cycling. Okay, hadn't done this, hadn't looked ahead. Got ahead of myself. All right, so cross-bridge cycling. As long as that calcium is there, you can have contraction. But you've got to have something else. You've got to have enough ATP. Okay, in order for actin and myosin to bind, you've got to have energy. That takes energy for actin and myosin to bind. So when you get fatigued, when you get tired, and you're like if you're holding something really heavy and finally your muscles start to shake and you just can't hold it anymore, you're getting fatigued. You basically depleted all your ATP. You've got to build that back up. So as long as you have calcium, as long as you have ATP, which during your normal everyday going about your business, you should have plenty of calcium, plenty of ATP. As long as you're well fed, you shouldn't have a problem with fatigue. As long as those are there, these cross bridges will form, and actin and myosin will bind, and you will get contraction. And this is called the power stroke. When myosin binds actin, and then you get that sliding filament, that movement, that's called the power stroke. All right. Now, there's a lot of steps involved in cross-bridge cycling, and they all have to do basically with ATP and what it's doing. All right, the first thing, kind of the cart before the horse. Let's assume that myosin and actin are already bound, okay? In order for actin and myosin to disengage, to detach, ATP has to come in to break that connection. So ATP has to come in order to break that connection. And again, kind of stay with me and think about when when might this occur? When might you not have any ATP and these things stay contracted? We're getting there, but kind of be thinking ahead if you can. All right, so ATP has to come in to disengage this. Now, here's actin, here's myosin. You've got to cock it. It's got to be ready to go, so it's got to be cocked so that it can cycle and actually do the power stroke. In order to cock the myosin head, again, it takes ATP. Let's see, I've got a picture. All right, so let's just start. Here is our cross bridge, okay? Let's see. Let's start down here because this is where I kind of told you we wanted to start. I'm going to start right here. We've already stroked, okay? Now we've already done the power stroke. ATP comes in this way. They always start with one. I like to start with one down here. To me, it just makes more sense. If it makes more sense to you to start up here, by all means do that. But for me, it makes more sense to start down here. So ATP comes in and causes that cross bridge to break. Okay, so you have ATP. Now you'll see your myosin head's not in the right position. You've got to get that head cocked so that you can have another power stroke. So what happens is you cock the myosin head. And you do that by ATP hydrolysis. Think of that word as hydrolysis. That means to cut with water. ATP is basically adenosine triphosphate. Adenosine diphosphate, ADP, this has 3 P's, this has 2 P's, and there's actually one called adenosine monophosphate. It just has 1. What happens is you hydrolyze or use water to basically cut off an ATP, or cut off a phosphorus, an organic phosphate right there. So you have ATP hydrolysis, which basically converts ATP to ADP. And whenever you break a bond, you release energy. Going back to Unit 1, we talked about chemistry. That's bonds. All bonds is stored energy. Remember potential energy and kinetic energy? Well, potential energy is the energy stored in that bond. So when you take hydrolysis, water, lysis, and break that bond, you release energy. Okay, so it takes energy to cock that head. Think about it. If you want to cock something, it takes some work. So you cock the head. All right, now it's ready. Calcium has come in, opened up that myosin binding site, and you get binding. Okay? Now the next step, the power stroke, basically ADP and the inorganic phosphate kick off. And when they kick off, you get the power stroke. Now you've got that binding again, ATP comes in, disengages, takes work, disengages that cross bridge formation. You hydrolyze ATP to ADP, cock the head, it binds, you get the power stroke. And this just goes around and round and round and round and over and over and over. Remember the little video just showing you like this, doing over and over and over and over and over again, as long as calcium and ATP are available. All right, let's see, do I want to show this or not? Hang on a second. All right, so here's our thick filament, here's our thin filament. We're blowing it up, and what we're doing is we're trying to take these Z lines and bring them together. So we're sliding our filaments over each other by alternatingly binding and releasing actin and myosin. Yeah, you guys get the picture. It's just the same thing over again. All right, what else? Okay, so as long as those myosin binding sites on actin are exposed, as long as calcium is bound to troponin, as long as troponin is keeping tropomyosin shapes changed, you will get this cycle. So every time you get a cycle, every time you get a power stroke, and every time actin and myosin bind, that sarcomere will shorten. And that's called the sliding filament theory. Those thin and thick filaments sliding past each other. Now, this cross-bridge cycling ends when you get rid of calcium. And so you get rid of calcium by actively transporting it. That means using ATP, using a pump, back into the sarcoplasmic reticulum. You take the calcium out of the cytoplasm, put it back in the sarcoplasmic reticulum. That means it no longer binds troponin. That means troponin then changes shape again. The tropomyosin changes shape again. The tropomyosin then covers the myosin binding site on the actin. So therefore, actin and myosin can no longer bind. You no longer get contraction. All right, so let's go back. So we've contracted the muscle now. So in order for it to relax, let's just kind of go back and see what do we need to do to stop it so we can relax that muscle. We got to stop the nerve impulse. Okay, so don't send out an action potential anymore. There's no more stimulus. Stop the stimulus. Okay, we got to get rid of the acetylcholine, either by breaking it down using acetylcholinesterase or diffusion. We got to get the calcium out of the cytosol, back into the sarcoplasmic reticulum. And now actin and myosin can't bind anymore. All right, so now we have relaxation. All right, so this is what I was kind of telling you guys to think ahead of me. Kind of guess what I'm thinking. You heard of rigor mortis, okay? Mortis means to die. Rigor is basically stiff, so it's called the stiffness of death. And you guys have heard of rigor mortis. It happens, 3 hours to about 3 days after you die. And that's, you just get basically stiff as a board. Okay, and why they call somebody that's dead a stiff? Because they really are stiff. And what happens is basically you have quit breathing. Okay, you're no longer breathing. And if you're no longer breathing, you're no longer getting oxygen. And I know you guys are so tired of hearing this little word, but I'm going to do it again. Cellular respiration. If you still don't know what I'm talking about when I use the word cellular respiration, you have got to go look it up. I want you to know it. Cellular respiration is when you use glucose and oxygen and go through a bunch of steps to make ATP. All right, so you're dead now. I mean, you may have just eaten a huge meal, so there may be plenty of sugar available, but you're not breathing anymore. There's no oxygen, so there's no ATP being formed. Okay, you've quit breathing. So what happens is now you have basically gotten rid of your ATP. Okay. So remember when actin and myosin bind, what do you have to break that bond? ATP, right? And those are going to stay bound as long as calcium is around as well. What do you have to pump calcium out? ATP. So what happens when you die, you don't have oxygen anymore, there's no ATP production, and that allows actin and myosin to stay bound and to stay contracted. They're fully contracted, and so you get that really stiff rigor mortis. It usually occurs about 3 hours after you die. It kind of maxes out about 12 hours. And then somewhere between 12 and 72 hours, you'll start to relax. And the reason you relax is not because you started breathing again, it's because you are decomposing. Basically your muscle, the protein is breaking down. So you're starting to decompose, and now those actin and myosin are just becoming disengaged because basically protein finally denatures or breaks down over time. And so you're starting to decompose, and then you will relax. So that's rigor mortis. So a reason I bring that up is you got to think about oxygen, ATP, and what all is going on in those cycles. I know it's a lot of information. Please don't freak out. We're going to get there. Just stay with me, and I hope this helps, and I will see you in lecture.